Hello everyone. In this section, we will talk about microbial taxonomy ranks. After learning this section, you should be able to outline the general scheme of microtaxonomy hierarchy. There are more than 100,000 microorganisms in the world. The diversity of microorganisms has always presented a challenge to us. We must classify all kinds of microorganisms if we want to know and study them. First, we should know what is microbial taxonomy. Microbial taxonomy is the science of classification according to the kinship and evolution of microorganisms. It comprised three distinct but highly interrelated parts that include classification, nomenclature, and identification. Classification means the arrangement of organisms into groups based on mutual similarity or evolutionary relatedness. Nomenclature means the assignment of names to taxonomic groups in agreement with published rules. Identification is a process of determination of the particular taxon to which a particular isolate belongs. In this class, let's get to know the classification. First of all, we should understand the general units of microbial classification. Microorganisms are placed in taxon. Taxon means levels of taxonomic rank in taxonomic hierarchy system. In this system, each organism in descending order is assigned to a kingdom, phylum, class, order, family, genus, and species. Each taxon has a specific suffix. For example, Acia denotes family, but Alas indicates order. They are the specific suffix of family and order. At the same time, if the taxonomy of this taxa is not sufficient to reflect the differences between certain taxa, the sub, super, tribe levels can also be added. The basic taxonomic group is a species. The most fundamental definition of microbial species is a collection of strains that share many stable properties and differ significantly from other groups of strains. Each species is assigned to a genus. A strain is a population of organisms that is distinguishable from at least some other populations in a taxonomic category. It is thought to have descended from a single organism or pure culture isolate. For each species, one strain is designated as a type strain. It is usually one of the first strains studied, and often is more fully characterized than other strains. To be recognized as a new species, genomic, metabolic, morphological, reproductive, and ecological data must be accepted and published in the International Journal of Systematic and Evolutionary Microbiology. Until that time, the new species name will appear in quotation marks. Also, There are several classification units below species. For example, subspecies, variety, form, and strain. Okay, we have learned the general scheme of taxonomic hierarchy. Let's see an example. Shigella falconeri, serotype 2A, strain 2457T. It is one of the strains in Shigella falconeri. It is assigned to bacteria. This is the domain level. Protobacteria. This is the phylum level. Gum protobacteria. This is the class level. Enterobacteriales. This is the order level. Interbacteriaceae. This is the family level. Shigella. This is the genus. And Shigella falconeri. This is the species. In this example, the strain is placed within higher taxonomic ranks. This is a word list for you. That's all. Thank you.